This is Bernard Paulson. I am going to discuss the material in chapter 4 in the book shown. In the first 3 lectures from this book, we have learned about mathematical modeling, the motivation for carrying it out. We have reviewed basic chemical kinetics, and we did detail the dynamic simulation procedure. From here on out in the course, we will apply this procedure over and over again to increasingly more complicated networks whose dynamic states we desire to characterize. We will start in this lecture by looking at very simple chemical mechanisms, set up the simulators for them, carry out the simulations, and discuss the results. First, I want to review some of the basic properties of chemical reactions that come up as you build these dynamic simulators. First is stoichiometry. Stoichiometry, in a way, is an accounting procedure where you count the molecules that participate in a reaction and those that are formed in the reaction. The stoichiometry is described by the stoichiometric coefficients that are integers, and they do not change. They are constants for a given reaction. The second property that we need to be aware of is related, I should say, to thermodynamics. There are many thermodynamic properties that we learn about. The one that is relevant here is the Gibbs free energy and the net change in the Gibbs free energy between the reactants and the products. This change determines the equilibrium constant. As shown in this slide, the equilibrium constant is a function of pressure and temperature. It is condition dependent. In most cases, in biology, we deal with a pressure of 1 atmosphere, and I should say that the Gibbs free energies associated with biochemical reactions are not typically a strong function of pressure, unless there are gas phase molecules involved. The equilibrium constant, on the other hand, can vary notably with temperature. In most cases, we deal with isothermal situations. If you are looking at human applications, well, the temperature will be 37 degrees unless the subject being studied has fever. In bioprocessing, for bioprocessing applications, most of the time we are dealing with temperature-controlled bioreactors. So for most practical purposes, for most situations, the thermodynamics don't change that much once you determine what they are. Thermodynamics determine the relative rates of reactions in the forward and the reverse direction. And this is, again, determined by basic thermodynamic properties. On the other hand, as we discussed in lecture 2, the absolute rates of a reaction vary. This is where enzymes come into play, and they can accelerate reaction rates by virtue of bringing the reactants into the right proximity with one another. As we pointed out in lecture 2, the shape of the surface of an enzyme molecule is encoded in the sequence of the gene, and of course the sequence can change, and therefore the kinetics can change. The absolute rate is therefore a biological property, unlike the first two. Essentially, all biochemical reactions are bilinear. What this means is that two different molecules, two different reactants, bounce off one another to form a joint entity. This could involve the formation of a covalent bond, or this could involve simply an association between the molecules. Here's an example of associations. This is the assembly process of hemoglobin that I mentioned earlier, where an alpha chain binds to a beta chain to form a heterodimer of alpha and beta globins. Then two of these heterodimers bounce off of one another, and if they bind, form the heterotetramer. Another example shown here is a stretch of DNA and a group of transcription factors that are protein that bind to the DNA. They encounter the DNA and bind to it, and once they are in the bound state, their properties are different. These are just some examples of bilinear reactions in biology. With that background, we are going to set up a few dynamic models for a few cases studied in chapter 4. The simplest case of all is a reversible linear reaction, shown here. A slightly more complicated situation is a bilinear reaction, where we have two reactants, as I just discussed, reacting to form a third molecule. These are the two fundamental types of reactions. We will then consider many of them occurring at the same time. I will first talk about a simple set of linear reactions as shown here. I will hasten to add that this is not interesting chemically, because if you look at this carefully, there is no change of elements in any of these molecules. These are simple rearrangements of the same molecule. So there is really no chemistry or chemical reactions involved in here. Nevertheless, we see an abundance of examples in the literature and in textbooks that simply draw out reactions like this. In reality, as I stated before, essentially everything in biology is bilinear. And we might be studying reaction mechanisms like this one, where we start out with a bilinear reaction to form this molecule X3 that then can break down into X4 and X5. This turns out to be a preamble to an enzymatic reaction mechanism. We will discuss those in the next chapter. And those will arise when, say, X2 and X4 are the same molecule. The enzyme before and after the catalyzed reaction. We will start simple with the reversible linear reaction. We need basic quantities for our simulation, and they are the stoichiometric matrix and the vector of reaction rates. These are shown in this slide. Let me briefly describe the stoichiometric matrix. Every row in that matrix corresponds to a compound, and there are two of them here. And every column corresponds to a reaction, and there are two of them here. So let's take the first column that here corresponds to V1, the forward reaction. In the first row of the matrix, I have a -1, which means that one molecule of X1 is consumed by this reaction. And I have an entry of 1 in the second row of the first column, which says that compound number 2 is being produced by reaction number 1. And the second column has the same information about the reverse reaction. The vector of reaction rates is very simple. We just line up the reaction rates, and they have to be in the same order in the flux vector as the columns appear in the stoichiometric matrix. And you can pick any order you like. And then we simply substitute in, for every entry in the flux vector, mass action kinetics as shown in this slide. So V1 is a function of X1, and that function is simply a rate constant times X1. The reaction rates are shown at the bottom of the slide. In an equilibrium state, V1 and V minus 1 are exactly equal in magnitude. If I equate V1 to V minus 1 and rearrange the equation a bit, I can show the equilibrium constant. The equilibrium constant, by definition, is the concentration of the products divided by the concentrations of the reactants at equilibrium. And we see here that that is simply the ratio between K1 and K minus 1. I will point out that the units on K1 and K minus 1 are the same. They are reciprocal time, and therefore the equilibrium constant for a linear reaction is dimensionless. All we need to do now is to introduce these quantities into the dynamic mass balance. And that, of course, is the equation at the top of the slide. We put in the stoichiometric matrix and the flux vector. When the reactions are linear, I can actually factor out all the rate constants as shown in the second line of this equation. Now I have a matrix filled with rate constants, and I have the vector of concentrations multiplying it. This happens to be the Jacobian matrix in this case, and it's obtained without any linearization. It is exact. We are not going to study properties of Jacobian matrices in this class. We can now look at these equations, and from that matrix equation, I can actually write out the individual differential equations as shown in the slide. So dx dt becomes equal to minus 1 times k1 x1 plus 1 times k minus 1 x2. And this is simply the inner product of the first row in the stoichiometric matrix and the flux vector. I do the same thing for dx dt, and I get the equation shown. What I'm noting in this block of the slide showing these two equations is that the dx1 dt is equal to the negative of dx2 dt. And this is, of course, because every time x1 reacts, x2 is formed. Therefore, these two derivatives are the same and tied together. And in fact, if I sum up the two equations and I take the derivative of x1 plus x2, that will be 0. So when a derivative of a quantity is 0, it means that that quantity is a constant. So this is simply a statement of the fact that x1 plus x2 is a constant, denoted by the letter A in this slide. I should also point out that when you look at the stoichiometric matrix, you see the rows are the same, just pointing in the opposite directions, and the same thing is true with the columns. This means that the rank of this matrix will be 1. You may want to refresh your memory from your linear algebra class about the rank. But the rank tells you how many independent linear equations are found in the matrix. And in this case, the answer is 1, which in turn means that the two quantities, x1 and x2, are not independent. And that is, of course, what the equation at the bottom of the slide shows. We call an equation like this a conservation relationship or a conservation quantity. Okay, so these are the equations, very simple. I can populate the differential equations, the dynamic mass balance with the quantities I have specified for this particular system, and I can simulate. And I know ahead of time that this is a one-dimensional dynamical system. So on this slide, I show a simulated response. I am going to pick the forward rate constant to be 1 and the equilibrium constant to be 1/2. I can pick these numbers myself when I make up simple examples like those shown in here. I cannot, of course, do that when I deal with a real system. This plot here shows the dynamic phase portrait for x1 and x2. The initial conditions I have chosen is 1 unit of x1 and nothing of x2. So basically, we start out with the reactant and we watch the reaction unfold. And in the phase plane, I simply see a straight line from the initial condition to the equilibrium line. So the equilibrium line is characterized by the ratio of x1 to x2 in the equilibrium state. And that is that slope, that is that line with a slope of 1/2. I can also plot the dynamic response, where I show the concentrations as a function of time. And that is shown in this insert. So x1 starts at 1/2 and drops down to 2/3. x2 starts at 0 and goes up to 1/3. So if I ratio 1/3 and 2/3, I get 1/2. So that would be the ratio of x2 to x1 in the equilibrium state. Note the time scale in this graph. All the dynamics are over in 1 to 2 time units on this scale. And this relates directly to the numerical value of K1 that is shown. K1 basically specifies the time scale that characterizes the response of this reaction. So there you have it. Easy to solve, easy to graphically display. Now let's talk a little bit about post-processing the solution. We can form pools here or aggregate variables. And what this represents is simply a variable transformation. Most of you have probably had classes on physics where you change coordinate systems. Going from a stationary to a moving coordinate system is, for instance, something you see often when studying the physics of motion. Forming pools or aggregate variables from the concentration variables is the same idea. I am just switching my coordinate system and looking at different variables. The first pool I'm going to pick here is shown in these equations. I'm going to put a row into the pooling matrix of 1, comma, minus 1 over the equilibrium constant. So when I multiply this row times the vector of concentrations, I get x1 minus x2 over k equilibrium. As this goes to 0, as shown, I would have x2 over x1 go to the equilibrium constant. So when P1 is 0, we are at equilibrium. When P1 is non-zero, we are out of equilibrium, and therefore we can call P1 a disequilibrium variable. We also see that if P1 is positive, the reaction proceeds in the forward direction. If P1 is negative, the reaction proceeds in the opposite direction. So I have simply transformed two concentration variables into a lumped representation, and that lumped representation tells me how far from equilibrium I am. The second pool I am going to pick here is x1 plus x2. And that is formed simply by placing a row in the pooling matrix of 1, 1. And when I multiply a row vector of 1, 1 times the concentration vector, I get x1 plus x2. In this case, I know that's a constant, and this pool simply represents the total mass in my system at any given point in time. So I have moved away from my original concentration variables into these pools by post-processing the solution. And these two pools simply mean different things to me. They measure distance from equilibrium, and they measure the total mass in the system. A third quantity we will see appear throughout this class is the state of a pool. So in this case, P2 is the total mass in the system, and if I take the ratio between x1 and P2, I see what fraction of that pool is in the form of x1. We will see this many times. For instance, with macromolecules like protein, there may be a total amount of protein present, but it can take on many different forms. And it's useful to know what fraction of the protein is in a particular state. So there we have it. Pooling is represented by a matrix that maps concentrations into the pooled variables. We decide what's in the pooling matrix. And in this case, I can take my solution for x as a function of time, multiply it by the pooling matrix, capital P in this case, and I get the dynamic changes in the aggregate variables or the pools. This matrix equation is detailed below with the two rows in it that I described on the previous slide. I can now add the time response of the pools to the previous representation of the simulation that I showed earlier, and that is panel C in this slide. Now I see that the dynamic response of P2 is time invariant, it's just a constant, it stays at unity, just as I knew, whereas P1, my distance from equilibrium, starts at 1, far away from 0, and decays to 0, which is the equilibrium state. And in the phase portrait, that corresponds to the point on the equilibrium line. So here you have it. This is the complete dynamic characterization of this very, very simple system. It is almost too simple to spend so much time on. However, I do so because all the fundamental concepts that we need to have in more complicated situations are encountered in going through this simple example. This slide just briefly shows rewriting of the net reaction rate to show you that the distance away from equilibrium, that second pool, is a part of that rate law. And K1, the magnitude of the forward rate constant, tells you how quickly the reaction responds to that distance away from equilibrium. Just a further interpretation or deeper understanding of pool number one. We now move on to a slightly more complicated situation, the reversible bilinear reaction. In this case, we have a forward rate that is bilinear. V1 now is a rate constant K1 times the concentration of X1 times the concentration of X2. The reverse reaction is simply a linear reaction that is a rate constant K minus 1 times the concentration of the product X3. Note that V1 is a bilinear equation. Also note that the units on K1 and K minus 1 will be different as detailed in lecture number 2. I can form the stoichiometric coefficient for, the stoichiometric matrix for this reaction. It is shown here. The first column will correspond to V1, and I have minus 1 in the row for X1 and minus 1 in the row for X2 because they both disappear in this reaction, 1 molecule of each. And in the third row for X3, I have 1, which means that 1 molecule of X3 is formed, and the second column representing V minus 1 is exactly the opposite. The flux vector is formed by simply populating it with the mass action rate laws shown above in the red box. Note that I have to order the elements in the flux vector in accordance with the way I order the columns in the stoichiometric matrix. The ordering is arbitrary. I can pick any order, but it has to be consistent between S and V. Now I have what I need for my dynamic mass balance. Of course, I now need to specify the rate constants and the initial conditions to carry out a simulation. Just want to look a little bit more at these equations. The differential equation for X1, as before, is minus V1 plus V minus 1, and I can now substitute in the rate laws there. And I have a bilinear term. This means that the differential equations are no longer linear. I also note here that I can look at the right-hand side of this equation and determine it is the negative of the net reaction shown. By rearranging this equation a little bit, factoring out K1, I can get at what the equilibrium constant is, as shown in this little insert. The equilibrium constant here, as before, is the forward rate constant divided by the reverse rate constant. It is also expressible in terms of the concentration at equilibrium, namely the concentration of the product divided by X1, the concentration of X1 and X2 multiplied together. Note here that the equilibrium constant has units. It'll have units of 1 over concentration. If I write out the differential equations for X2 and X3, I see that they are the same as the differential equation for X1. And this is immediately apparent by looking at the stoichiometric matrix and realizing the rows are all the same. There is only one row in there. And you also see the columns are the same. And again, this means the rank is 1, and I will only have one dynamically independent variable in here. This in turn means that I have two conservation variables, and you can immediately see them by looking at the rows in the stoichiometric matrix. If I add up the first and the third row, I get 0. If I add up the second and the third row, I get 0. and that corresponds to these two conservation quantities shown in the red box towards the bottom of this slide. As with the linear reaction, reversible reaction, I can form aggregate variables here. I can form a disequilibrium variable as shown in here that is analogous to the one I had for the simpler case. The big difference here is that P1 as defined there is a nonlinear function, and I cannot describe it as a row vector times the concentration vector. To do that, I have to linearize this equation. I am not going to discuss linearization in this lecture here, but how that is done is found in the text, and I have some extra slides at the end of the lecture for those who are interested. I would say that anyone taking this class after having linear algebra and differential equations should know that linearization is a Taylor series expansion of the nonlinear equation around a fixed point of interest. The second two pools I can define here are the conservation variables that I mentioned earlier. And it's easy to understand them if you write out the chemical reaction in more detail as shown here. Here I am calling the two reactants C and A. And C and A combine to form a joint molecule Ca. But upon inspecting this equation, I see, for instance, that the moiety A is either free by itself in the form of X2 or bound in the form of X3. And the summation of those two has to be a constant because no A is entering or leaving the system. And that will be pool 3. And the same thing is true for the other reactant. It doesn't disappear. It's either free or in the bound form. I can put this on my simulator and calculate. To do that, I pick these values of the rate constants shown here. K1 and K minus 1 is being unity. The simulation to the left shows a case where I start X1 out at 3 and X2 out at 2 and have no product present in the beginning. So X3 starts out at 0, then I just simulate and it goes to equilibrium. And you can verify, and it is detailed in the text, that with an equilibrium constant of 1, you can solve for the equilibrium concentrations there, knowing that X1 and X2 were at these initial values. The graph to the right shows the response of the pools. As expected, pool 2 and 3 don't change. They're conserved. There's no change in time. The disequilibrium variable starts out at somewhere about between 4 and at 6, looks like. Yeah, starts out at 6, 3 times 2, and decays to 0. There's a little insert shown there, or a little, I should say a second line shown there, which is the linearized version of this pool, and that is discussed in detail in the text and not here. So setting up simulators, dynamic mass balance simulators for chemical reactions is easy. You can formulate the stoichiometric matrix and put in the mass action based rate laws into the flux vector, and you can calculate. We will now consider slightly more complicated circumstances. We will start out with this series of linear reactions. We're going to have, in this toy example, a reversible reaction followed by an irreversible reaction that is then followed by a reversible reaction. And in your mind, as we go through this, we should be thinking about the two reversible reactions as being fast, and the second one being fast or slow. We're going to look at the effects of the relative magnitude of K2 that characterizes that second reaction and the others. I do this to begin to develop an understanding of multi-scale, multi-timescale dynamics. When you have one reaction, there's only one timescale. When you have three reactions, there are three timescales. And in this case, we're going to look at the effect of separating these timescales and just looking at the effect on the dynamic response. The stoichiometric matrix here is a little complicated, a little more, I should say a little more complicated than before. I have 5 reaction rates in here, therefore 5 columns. I have 4 concentrations and 4 rows. I am drawing dashed lines around 2 blocks in this matrix that look exactly the same as for the first case we studied. This simply corresponds to the 2 reversible reactions. And then you see that V2 is actually a coupling variable between the two of them. What I mean by that, if K2 goes to 0 and that reaction becomes infinitely slow, the other 2 reactions will become dynamically decoupled. So the coupling between these 2 reactions, the dynamic decoupling between the 2 reversible reactions is going to be dependent on the magnitude of the rate constant K2. I can populate my flux vector with the mass action rate laws as shown in here, and I can substitute this into the dynamic mass balance. And as before, in this case with only linear reactions, I can factor out the rate constants if I want to, and form the Jacobian matrix as shown below. Again, that's not something of primary interest here. I can take this stoichiometric matrix and this reaction vector, this flux vector, and simulate them. And of course, to do that, I need to specify the initial conditions as well as the numerical values for these rate constants. And an example of that is shown here. In this case, I am just starting out with x1 and all the other concentrations are 0. I have chosen the rate constants, forward rate constants for the 3 reactions to be the same, and I have picked the equilibrium constants to be unity. You see that in the beginning, x1 is converted to x2 and x2 builds up. Almost as soon as x2 starts building up, x2 starts being converted to x3, that then in turn equilibrates with x4. All of these reactions are happening on the same time scale. So this is the most complicated dynamic response that I can get from these 3 reactions. They're all proceeding on the same time scale and all dynamically interacting. Now, if K2 is 0, then I would have 2 independent subsystems in here, and I would pick a pooling matrix based on my study of the first example in this lecture. But if K2 is not 0, this becomes a little more complicated. I will no longer have a mass balance between x1 and x2 on one hand and x2 and x3 on the other hand. That will be broken. On the other hand, I will have the total mass being a constant. So x1 plus x2 plus x3 plus x4 has to be a constant. And that is represented by the bottom row, the 4th row in the matrix on the right. In that matrix, the first and the third row actually represent the, the, the disequilibrium quantities for each one of those reactions. The second row represents just the coupling variable x2. So in trying to form pools for this system, I have in this case chosen P1 and P3 as being disequilibrium variables associated with the two reversible reactions in here. I have chosen P2 to be the coupling variable because it drives the second reaction. And P4, I know, will be a conservation variable. If you do take the stoichiometric matrix shown in the previous slide and calculate its rank, you will find out it is 3. So there are 3 dynamic degrees of freedom here, not 4. If I simulate, I get what is in this slide over here. I should say if I simulate again. And in this case, I have changed my numerical values for the rate constants a little bit. And this is shown in the red box to the bottom left. I still pick K, the equilibrium constants as being equal and equal to 1. Now I have picked K2 to be 5 times slower than K1 and K3. And I also pick a numerical value of 1 for K1 and K3. But K2 will now be 0.2. If you look at panel A, you see now a much simpler dynamic response than before. And before is the case when I had all the forward rate constants being the same. Here we see in the beginning that x1 and x2 rapidly approach one another, and then they move in a ratio of roughly 1-to-1 on a longer time scale. Notice the time scale now goes out to 25. Before it was much shorter. This longer time scale is now governed by K2, that is the reaction that slowly drains the equilibrium state formed between x1 and x2. As soon as x2 is converted to x3, x3 very quickly equilibrates to x4. And therefore, the concentrations of x3 and x4 kind of move in tandem, very close to being equal in value and very close to being in equilibrium. They are not strictly in equilibrium, they are in quasi-equilibrium. Notice also that when you look at this reaction mechanism, you know what the end state will be. Because of the irreversible reaction in the middle, as time goes to infinity, there will be no x1 or x2 left. All the mass will be in the form of x3 and x4, and they will be equal in magnitude at the end because the equilibrium constant is 1. And in fact, since the total mass in the system is always unity, the end concentration for those will be a half. And you can see as time goes on, those converge to a half. Panel B shows my post-processing of the dynamic responses using the pooling matrix where I did not use the coupling variable. And you see in this case all the pools are moving. P1 and P3 very rapidly go into what we call a quasi-equilibrium state. It's not exactly at equilibrium because it isn't perfectly zero, but it's close to being zero. And then P1 and P2, which are the total mass variables associated with reaction 1 and 2, these are indexed as P2 and P4. And you can see how one drops and the other comes up. In the end, everything is in the form of x3 and x4. So this definition of pool 4 has to go to unity in the end. Panel C shows my different definition of the pooling matrix. Here you see that P4 is a constant at unity. It is the total mass in the system. The two disequilibrium pools, P1 and P3, behave exactly the same as in panel B. And then P2 is now my coupling variable that very rapidly moves towards a half, a little bit above 0.4, and then dynamically decouples the reactions together on this longer time scale. And so it's that longer time scale that drives basically the draining of the X1 plus X2 pool into the X3 plus X4 pool. And those two pools are very clearly seen in panel B. So you see in this example that there are rapid equilibrations of two reactions. And then there's a slow reaction that is draining one pool and filling another. The dynamic response in panel A looks a lot simpler than before. Just thinking about this example, we see the possibility for temporal decomposition, namely two rapidly equilibrating reactions and one slow coupling variable driving the drainage of one pool and filling another. Again, these are concepts we will see over and over again, and sometimes a little more subtle and complicated than shown here. The final example in here is one of two coupled bilinear reactions, as shown. Here we will have 5 compounds and 4 reactions. And looking forward to the next chapter, I should state that if X2 and X4 are the same variable, this is the reversible form of the Michaelis-Menten mechanism. Namely, X2 is the enzyme that binds to the substrate, X1, to form an intermediate complex called X3. The reaction then takes place on that complex, liberating X5 as the product and regenerating the enzyme X4, which would be the same as X2 in the case of enzyme catalysis. So this is the last example in this chapter, and it's a segue into enzyme kinetics covered in the next one. The stoichiometric matrix here is easy to form: 4 columns for the 4 reactions, 5 rows for the 5 compounds involved. Here I draw 2 dashed boxes around 2 blocks in this matrix, and you will identify them as being identically the same as the stoichiometric matrix for a single bilinear reaction. I'm also putting a dashed line around row 3 in here. Okay, so these are the block diagonal terms, and then I draw a dashed line around row 3 in here because X3 is a participant in both blocks. X3 is a coupling variable. In this case, unlike a coupling link, like in the previous example, this is a hard coupled variable. You cannot change this coupling at all because the concentration of X3 will drive all these reactions the same way. Then I can write out my mass action kinetics for every reaction and populate the flux vector. I can then put these into my dynamic mass balance, and I can now simulate. And of course, to do that, I need to specify the numerical values for the rate constants as well as the initial conditions. You should be getting the hang of this now. It's always the same process, and I'm following exactly the workflow outlined in chapter 3 or lecture 3. I want to look at this matrix a little bit, the stoichiometric matrix. You will see that the first 2 rows are the same and the last 2 rows are the same. And you will in fact see the third row as a summation of the first row and the last row. So there are three dependencies amongst the rows in this matrix, and the rank will therefore be 2. You can also see by looking at the columns that the first 2 columns are the same and the second 2 columns are the same. So there are only 2 independent columns. So the rank is 2, and this means the degree of freedom is, the dynamic degree of freedom is 2. Since I have 5 concentrations, this means that I will have now 3 conservation quantities. Here is the definition of conservation quantities. These would be 3 pools that I can define by this matrix over here. As I did for the bilinear reaction, I can trace out the moieties going through here. For instance, if I look at the first row in here, it corresponds to a conservation of a moiety A, which in this case starts out in X1, ends up in the complex X3, and then is liberated in the form of X4. So the summation of X1, X3, and X4 has to be a constant. And the same for moiety B and moiety C. So I can form 3 pools from this, and I can also form disequilibrium pools for the 2 bilinear reactions as I did before. And now I can simulate and I can post-process. So here's just one simulation where I start out with the same amount of X1 and X2. I assign them a value of 3, and all the other concentrations are 0 in the beginning. You see X1 and X2 move exactly the same way because there is one of each consumed in the first reaction. You see X3 build up, then you see X3 start to fall apart and to form X4 and X5, and of course they behave identically because for every molecule of X3 that falls apart, I get 1 X4 and 1 X5. The numerical values picked for this are shown below. Again, the rate constants, forward rate constants of K1 and K3 are picked to be unity, so the time scale for the dynamics is unity, as shown by the x-axis in these plots. The panel to the right shows post-processing of the time profiles for the concentrations. We now see 3 pools that are conserved, P3, P4, and P5. And then we see the 2 disequilibrium pools, P1 and P2, decay to 0 over about 2 to 3 time units on this time scale. Here's an example phase portrait to interpret this reaction system a little bit. Here I'm just plotting the 2 pools, P1 versus P2, the dynamic responses. And for the traces shown in here, I have varied the forward rate constants. For trace B in there, you see I move from the time 0 in a straight linear fashion into the origin, which is time infinity. Both pools are fully gone to 0. In this case, the time constants are identical for the 2 forward reactions. In panel A, I have made pool 1 faster than pool 3. So you see pool 1 shoot across horizontally to the y-axis and then moves vertically down the y-axis as pool 2 relaxes. And trace C is the opposite case where I made pool 2 fast and pool 1 slow. So this is a use of a parameter portrait to look at variations in the numerical values of the kinetic constants. This foreshadows some of the things we might do later with simulation, where we change the relative magnitudes of key rate constants. So this is the end of the lecture, and there are a few summary bullet points to cover. First, we are simulating chemical reactions, and the key properties associated with chemical reactions are stoichiometry, thermodynamics, and kinetics. The first two of these properties are physical-chemical properties, while the third is biological. So living cells have to abide by the constraints of the first two while they can manipulate the numerical value of the third. All the simulators that we are going to build are of biochemical reaction networks where every chemical transformation has to abide by these constraints and will exhibit these three features shown in here. We showed in the lecture that the dynamic mass balances are very easy to form. You just form the stoichiometric matrix and you populate the flux vector with the mass action based kinetic rate laws. You need to be careful with the indexing to make sure that the columns in the matrix and the order of the rate laws in the flux vector are the same. You then simply simulate dynamically and then you graph. The primary solution can be productively post-processed in these cases by using aggregate variables. And these aggregate variables correspond to disequilibrium pools and conservation pools. And for fast-acting equilibrating reactions, you will have a dynamic motion that is typically too rapid to be of interest, and you can relax it. And you can relax those dynamics. If you remove dynamics like that, it means that the dynamic dimension of the description simplifies. We can see this through complicated mathematics, or often you can see this just directly in the graphical representation of the response. And I try to give some insight into that, particularly in the third example in this lecture. There are some extra slides in here for those that are interested in the more advanced topics of block diagonalization of the stoichiometric matrix, and linearization. I will not cover these topics in this lecture, but they are in the book. You are now ready to build more complicated simulators. The lecture illustrates the dynamic simulation process by using very, very simple examples. Again, the process in principle is always the same, the procedure is the same, the workflow is the same, but it gets more complicated in detail as the networks you're dealing with get more complicated. The stoichiometric matrix gets bigger, and the graphical representation gets more complicated. But in principle, it's always the same. So that concludes this lecture on simulating dynamics of simple chemical reactions. Thank you for your attention.